All right. So, hi, this is Navigating Nano, a podcast by the Materials Research Society at JSNN, the Joint School of Nanoscience and Nanoengineering between UNCG and A&T. And I'm Hilary Dimig. I'm a fourth year PhD student, and I have with me Dr. Lakeisha Tate. She got her BS in chemical engineering from the University of Alabama, a PhD in bioengineering at Rice, and postdoc of bioengineering at University of Washington. And now she's an assistant professor at UVA, University of Virginia. And she works on hydrogels and vascularization. And I'll let her talk a little bit more about that. So how this will go is I'll ask a few questions, and we're going to try to keep it pretty casual, and then I'll let the audience ask any questions they have. And then we have our last question is from the last speaker, and it's kind of a fun question. And then I'll get a question from you to ask the next week's speaker.
Awesome.
All right. So do you want to give just a little blurb about your research, just for anyone who doesn't know already?
Sure. So thanks for having me. So my research is focused on the development of biomaterials, mainly for vascular tissue engineering, but we're always open to other types of tissue engineering. I like to say that every organ system needs a vasculature for delivery of nutrients and transport of waste. So we're always open to working with people who are working with other tissue systems. But we focus mainly on synthetic materials for use as hydrogels in drug delivery and tissue engineering.
Cool. And what made you interested in research in STEM and in biomaterials specifically?
Oh, wow. So when I started at the University of Alabama, I had no idea what it was like to do research, probably like a lot of undergraduates, and I had no idea how to get involved in research. When I went for orientation, I managed to sit at lunch next to the person who was running the computer-based honors program. I had never even heard of the program, but just talking to her at lunch and learning what that program was about and how the students got to do computational research as part of their curriculum, I thought, wow, this is really interesting, and maybe this is something that I would want to be a part of. And she actually said to me, you know what, we still have one spot open. Would you like to participate in the program? And so when I started at Alabama, I started in the computer-based honors program. They do an accelerated course of teaching you programming in your first year, and then they expect you to do computer-based research throughout your time as an undergraduate. I managed to find a project in the chemistry department, which I thought was really interesting. Again, I had no idea what I was doing, but they booked me time on the supercomputer, and I got to study how this particular set of salts interacts with polyethylene glycol. And who knew I'd end up working on polyethylene glycol later, but working on that project sort of opened my mind to doing lots of different things. At one point, the grad student said, would you like to come in the lab and do some experiments? Oh my God, they trust me to touch things too. So I got to try a little bit of everything. And actually that work ended up in a paper, which I never even saw the paper until after I left and started grad school. But that was actually what started me working in any type of materials. And then when I went to grad school, I knew that I wanted to do something that was more biomedical focused. And the project that I chose when I got there just happened to be based on, again, polyethylene glycol, but we were using it for drug delivery. And so my interest in biomaterials from there just kind of exploded because I was working in one particular type of material, but I kept going to my advisor and saying, what if we made this? Or what if we tried this? And luckily she was the type of person who was open to all kinds of things. And so I got to make nanoparticles, I got to make polyurethanes, I got to make several different types of materials. And that just sort of cemented that this was the right thing for me to be doing.
Cool. So what was that transition from computational to wet lab like?
It felt abrupt, but I always liked labs, right? So I always liked putting my hands on something and trying different things. So for me, getting to go into the lab and actually do the experiments that sort of backed up what I was seeing computationally was just heaven. It was just the thing I really wanted to do. And so that's why I'm now much more of an experimentalist. We're sort of dipping our toes into maybe trying some computational stuff, but yeah, most of my lab is experimental because I just like doing hands-on work.
All right. What challenges have you faced in your research career, and how did you cope with them?
Oh, so many challenges. So just like everybody, you start doing something and you kind of hit a wall, right? So you have to figure out how to get around the wall, or whether you need to take a step back and figure out how to go in different directions. I feel like that's something that everybody experiences who's doing research, right? For me, I've bounced around a little. So I started as an assistant professor at Georgia Tech in their School of Chemical and Biomolecular Engineering. And it was a really large department, lots of students, lots of faculty, and it never quite felt like the right fit for me. So I think figuring out the environment that I wanted to be in was kind of my biggest hurdle. I moved into a position that was much more research oriented, and I worked in the vet school at Texas A&M, which people don't necessarily immediately make that connection with the vet school and regenerative medicine, but a lot of people are doing research in that area because, I mean, what happens if you need a horse who needs a new ligament? So it also seemed like a really good fit, and I did really enjoy my time there as well. And when the opportunity came to move to UVA, a smaller department, kind of intimate. Everybody knows each other. All the students hang out together. People live together, as well as work together, and it is just kind of the perfect environment, I feel, for the type of research that I do and the type of students that I like to work with in my group.
Yeah, I like that about UNCG too, and I'm sure people at A&T also feel that way. I did undergraduate research at the vet campus at NC State, and like we were looking at cow guts, and we used like bioreactor equations to look at cow guts. So
It's a big bioreactor.
Yeah, I thought that was pretty cool. So what is your philosophy as a mentor?
Oh, wow. I like to tell students that I'm available for as much or as little mentoring as they want. Every student's different, right? So I'm nosy, but I like to know what my students are doing. I probably walk over and poke my head in the lab at least once a day just to see who's doing what, who might need help, what's on the docket for this week, kind of thing. But I tell my students, if you want to come talk to me, if you can find me in my office, let's chat. If you're the kind of student who does not want to talk to me every day, that's also fine, as long as I have some idea that you're doing work. And if you want to bounce stuff off me, we can email. So I think that, mentoring has to be a little different for everybody, if that makes sense. I also mentor quite a few undergrads in my lab as well. So it can be a little sort of off the hip. Somebody will stop by my office and we can chat for a few minutes, or I find them in the lab and we can chat for a few minutes. We also have a weekly meeting where everybody gets to sort of air their grievances or get advice or whatever they want to do. So yeah, I tend to think that it has to be tailored to the specific person that you're trying to mentor.
What kind of mentorship did you enjoy when you were in grad school?
So funnily enough, the dean of the School of Engineering at UVA was my graduate advisor. So she's still mentoring me to this day. As a grad student, I always felt like whatever came up, I could go and talk to her. And so that, I think, shaped me quite a bit in the way that I mentor my students, because I always felt like if I had an idea or if I had a question, I had a sounding board. And, it's good to be able to talk to your fellow students, but when you feel like you can go to your professor and just open up about any concerns that you have or things that you like to try, it's a little bit sort of validating. It's that extra bit of validation that, oh, okay, maybe this isn't such a crazy idea and maybe I'm on the right track. And so she was really great at pushing me to try new things, and letting me do things that I came up with myself.
Cool.
Yeah.
All right. So this kind of ties back into what you were saying before about hitting a block when you're doing research, but what do you do to stimulate creativity when identifying new research topics?
I think it has to be talking to people, honestly. Um, when I have a new idea, it's easy to go down sort of the rabbit hole of, opening 27 tabs and having a new paper in every tab and trying to figure out, taking notes and figuring out all the things that I can try. But taking those notes and actually sitting down and talking to other people and saying, hey, I have this idea, I want to bounce off you. What do you think about this? There are people in my department who are very much biomaterialists the same way that I am, but there are people who are slightly more chemistry oriented who can tell me, yeah, that reaction is not going to work. And then there are people who work in different cell types who say things like, oh, well, maybe we could also try doing this together. And so I think that helps me most of all, is being able to sort of air it out and have somebody take a look at it and say, okay, I see where you're going with that, and maybe this makes sense, and maybe you could also add this piece or that piece.
Cool. And so my last question before I'll turn it over to the audience is, what advice do you have for current graduate students?
Try everything. And it sounds so easy to say. Like, I know you guys are busy, you have tons of stuff to get done, right? You're trying to focus on your specific research and trying all the things you can to make it work the way you think it should work. Mentor undergrads, go to conferences, go to workshops on mentoring, on writing, on doing presentations, all those things that you can usually find that people do that you think, oh, maybe I'll think about that later. Try it. You never know what's going to happen. I'll never forget my very first year as a graduate student. I've always been kind of the quiet person who's happy to stand in the back corner and just not be out front. But I knew as a graduate student, I was going to need to do presentations at conferences. That's just part of my training, right? So I volunteered to go to this small conference and enter a competition for presentations. I don't know what I was thinking. But I went to this conference. I was the youngest person in my division. So I was the only first-year graduate student. When I got up to give my talk, something happened to the computer and I couldn't see my slides.
No.
So I kind of panicked. But there was a guy in the audience, and he said, you can do this without seeing those slides, right? And I said, okay, sure. And I gave my presentation, and I sat down, and my heart was beating out of my chest. And later that day, they gave me the first place award.
Nice.
And I never could have imagined that would happen to me. But I kind of took a chance and tried something because I knew it was something that I wanted to improve upon, and it paid off. And so when I actually went to my first large conference and stared out at the audience, which was a much bigger audience than I was expecting, I felt okay giving that talk. And so, yeah, I would just say try any and everything. If you get invited to something, try it. If you come up with an idea and you want to mentor this student, or you want to go to a high school and talk to students about being in STEM, just do it. All those little things kind of add up to make you into the researcher or the mentor that you eventually want to be.
I actually have one more question. It's kind of niche question. So I'm like a lab grown meat nerd. I like to follow the news about it. Do you think there's any application for like the work that you do in lab grown meat?
Potentially. I mean, it's tissue engineering, right?
Yeah.
So if you need an environment to grow a specific type of muscle tissue, we have people doing musculoskeletal tissue engineering in my department, then yeah, definitely. I think that could be something really interesting. I actually have a former student of mine who's gotten very interested in plant tissue engineering, and so she's always asking me questions about the extracellular matrix and what if we could try this with hydrogels, and I'm like, let's try it. So yeah, I think that's definitely possible.
Okay. So I'll turn it over to the audience now if anyone has questions.
Yes.
So I think the talk you gave is awesome.
Oh, thank you.
And like I like what you work on. So I'm working on mimicking the blood flow in veins.
Okay.
So I heard you talking. You made a microfluidic device that kind of relates the blood-brain function.
Yes.
So I was thinking, I want to know in the details how you made that, and like to be specific, what environmental factors or other factors you had in your mind when you were fabricating the pieces, and what gave you the final device.
Okay.
The
So
factors.
I'll have to say first that I had a student who was most of the brains behind this particular experiment. I came to her and I said, I think it would be really nice if we looked at the blood-brain barrier and maybe figure out if we could make a model system to look at drug transport across the blood-brain barrier. And she, having never made a microfluidic device before in her life, she figured out how to design the device herself and how to make the device. So she was making PDMS, basically sort of lab on a chip type device. She figured out what requirements were necessary for the biomaterial matrix that would support her cell types. She figured out how to get the right amount of physiological flow to develop these cells into a vessel that could withstand the physiological shear. I can talk to you a little bit about exactly what she did and show you some of her results as well. But yeah, she did it all. I can't take too much credit for it. But it turned out to work really well. And she's actually gone on to work in industry where she got to do a little bit more of that type of development with microfluidic devices and tissue chips. So it worked amazingly well. But
Yeah, that's cool. So I would like to get connected with you, and like I can get some hang up like what you are doing now, because that's what I'm doing also.
Okay.
I'm thinking of a shear stress, and I'm also thinking of like pH and nutrient availability and some other things, and so I was thinking what. But I'm not from this background also, but I enjoy working here. So,
Okay.
so I was thinking like, maybe I'm missing something. So I would like to talk more with you again.
I feel like every graduate student has that thing where maybe I'm just missing the one thing that's going to make this work perfectly. But I can definitely keep in touch. We can talk about, again, what my student did. I can probably put you in touch with her as well if you're interested, because she can tell you exactly how she put the bits and pieces together. But yeah, that would be great.
Thanks a lot. It's a pleasure talking to you.
Thanks.
Yeah, I have a question. I think it's interesting that you said that I guess you don't really like the spotlight, but because you seem really charismatic, but I feel like, I mean, you are charismatic, but I feel like you're just like kind of really curious too, which is pretty cool. Because like you talked about how like you just got random opportunities with just being like, hey, what's up with this, like, and just talking to people. So that's pretty cool, and I guess that's something I'm kind of learning nowadays. Like I try to be at my desk a little bit more. I'm not a hermit, but like introverted, but I'm trying. So that's very, how do I say this, inspiring. But the question I had, since he asked the question about like kind of like neuroengineering, I want to, right now I'm kind of working with 3D printing, but with the added time dimension kind of 4D printing. And specifically for different applications, but really looking into like kind of like the neuro engineering aspect. And so I was just wondering, like, is there any like kind of like application for like materials that could like interact with kind of like the free energy of kind of like brain tissues and kind of like adapt to it versus just like kind of like regular drug delivery? Like, can you think of anything in the database in your brain to see like, all right, maybe something like smart biomaterials 4D printing could like have some sort of application, like kind of neuroengineering?
Yeah, so I know quite a few people who are doing neural tissue engineering with lots of different types of materials. People are doing cell delivery in sort of these really soft matrices to see whether or not if you inject cells from the brain, they can survive, sort of thing. Um, are you more interested in delivering things, or are you more interested in just the tissue engineering, so having a matrix, or?
Yeah, having a matrix and being able to kind of repair nerve tissues, either kind of like speed up the recovery or just like be able to repair it. Because I know like in the kind of like central nervous system doesn't really have as much capabilities as like the peripheral.
Right.
So I think I don't know, something like that can help. I'm still in the early stages, but I was like
Okay.
just kind of collecting some information.
Yeah, I think there's a lot of possibilities there. Some of those cells don't really have as much capability to repair themselves, or regenerate or proliferate or things like that. So people are using some stem cell technologies as well, in terms of, can you build a little bit of brain in a biomaterial to be implanted into the brain? There's definitely stuff like that going on. So I think there's a lot of possibilities in that arena. There's a professor in our department who does some neural tissue engineering, so I could definitely point you in his direction. And I can think of a few papers just off the top of my head where people are doing sort of that type of work. So absolutely.
Okay, and then he also mentioned the blood-brain barrier. Can
Yes.
you explain that like I'm 5?
I can actually. So the blood vessels around your brain have to be highly specialized because you don't want random things to get to your brain, right? So there's this network of very highly specialized blood vessels that is termed the blood-brain barrier, that have these very sort of tight junctions that strictly regulate what can get sort of in and out of your brain. Does that help?
Okay, that makes sense. So, pretty much you have to be very, like outside of like making sure that your material is biocompatible and can actually like regenerate cells and biodegrade. You have this blood-brain barrier
Right.
that's like what kind of like I want to say like the police, but like kind of like
Kind of.
okay.
Kind of, because your brain is very sensitive, right? So if any old thing can cross the sort of that barrier to get to your brain, then all kinds of things could be floating around in there, and that's definitely not what you want. So when people think about delivering drugs to the brain, a very hot topic in research is how do we get our drug to be able to cross the blood-brain barrier, because it is so strictly regulated. So in a way, it does sort of police what can go into your brain, and it's very important because otherwise, I mean, all kinds of things could get to your brain, and that could cause all kinds of dysfunction in the brain. Does that?
Yeah, okay, that makes sense.
Yeah.
So, okay, so, I guess, what type of, so the material has to match, kind of like have this kind of like ID or something to it for the blood-brain barrier, like almost like tricking it, almost.
Kind of. So there are some active transporters, which the cells use to let things through the blood-brain barrier. So if it recognizes this particular type of ion or something, okay, that can go into the blood-brain barrier. So sometimes people will try to design a drug that can maybe mimic the way some of those things are transported across the blood-brain barrier, because otherwise it is very difficult to get things across. So you can think of it as kind of being able to sneak past the police, maybe. That is the blood-brain barrier to get in. It's very tricky to be able to deliver things to the brain. But yeah. So some ways that people will get drugs into the brain. So if you have a brain tumor, sometimes they have to actually like open up your skull and try to take the tumor out, and then they will just put something in the cavity that can release drugs over time and then close everything up, because trying to get that drug into the brain otherwise is much more difficult. Okay.
Hi,
Hi.
my name is Sarah Lors, and I joined the Material Research Society this semester. I'm a second year Master of Science in Nano Engineering student, and my question is about your research with polymers that you discussed during the seminar earlier. Have you created any publications about polymers?
Yes. So we recently had our paper on the matrix stiffness with the lung study come out in microcirculation. I published a bunch of papers with my students while we were at Georgia Tech. So on the elastin mimicking peptide, I had a student doing work on stimuli responsive polymers, and he put out a lot of papers, just polymer synthesis type, polymer chemistry type papers. There's a bunch of papers from my PhD based on polymers that we made for nitric oxide delivery. So we did some polyurethanes there. We're currently working on a paper with the polyurethane that we've developed with Professor Dumas, as well as trying to get students started to think about how to publish their work that they've started in my group as well.
Okay, I'm working on a project about nanocellulose, and I would like to use some of your publications as a reference.
Okay.
Possibly.
Okay, sounds good. I can definitely help you find those.
Thank you.
Oh yeah, I have another question. Also seems like you seem like so you went to Georgia Tech versus like Alabama, right? Alabama.
So I went to Alabama for undergrad. I went to Rice for grad school. Georgia Tech was my first faculty appointment.
Okay, Rice is in Houston.
Houston.
Okay, and then now you're in Charlottesville. So I'm
I
guessing
bounced around all over the place.
right. So you switched between like kind of big city and like small, large college township.
Yes.
Does that kind of like because you seem like more like an intimate setting person, I guess, kind of like were the kind of cultures at these different, like, how do I say this? Georgia Tech's in a big city, Rice is in a big city. Charlottesville is a small town, intimate town. Alabama is a small, intimate town. Does that kind of like the kind of culture of like kind of the city you're in kind of like reflect what kind of type of lab you're in? Because you mentioned like Georgia Tech is like this big kind of like overwhelming department.
I mean, I think it depends, because even sometimes when you're in a big city, if you find like your space, it feels a little bit more intimate. Like you find like the neighborhood that you live in, the grocery store that you go to, and the places where you hang out. It doesn't necessarily mean I have to drive for 3 hours through Houston to find where I want to be, kind of thing. So I think it kind of turns out to be what you make of it. I don't mind living in a big city. I don't mind living in a small city. I grew up in a really, really, really small town in Alabama. So as long as there's more than one grocery store, it feels like a big city to me. So I bounced around to different places, and I can say that there's good in all of those places, and there are things I liked about being in all of those places. And I don't think you necessarily let the place dictate who you're going to be in that place. I think you have to find a way to dictate how you're going to adapt to that place, if that makes sense.
Yeah. Okay, yeah.
Because when my first big city, I moved to Houston as a graduate student, and it was completely overwhelming. But then when you kind of figure out and get into the groove of, okay, I know where to go if I want to find this, and I know where to go if I want to find that, and maybe if something's way out in the boonies or wherever, I can figure out how to get myself there, see how much I like it, maybe take a friend. We'll figure it out together. Yeah, it's nice to be able to explore, right? So,
Right.
But yeah, you get used to it.
Okay. All right. So there's no reflection on the city on kind of like the academic culture at all.
I mean, each academic institution is a little bit different in its own way, but I feel like it's more about the people for me. Everywhere you go, you're going to meet different people, but students are students, and a lot of them have like the same issues, and you end up talking about a lot of the same things. So yeah, there's similarities everywhere you go.
So I found you on LinkedIn.
Okay.
So are you active in LinkedIn? Should I message you there, or I can
You
email
can
also?
message me there. Sometimes I do go down the rabbit hole of LinkedIn. I'll get an email to say this person would like to connect, and then I connect, and then I look at their page, and then I spend like the next 30 minutes scrolling through to see what's going on. Yeah. So I wouldn't say I'm the most active person on LinkedIn, but if you message me, I will definitely see it and get back to you. So
That's
yeah,
great.
I will say somewhat.
Thank you.
All right, is that all the audience questions? Okay, so the question from our last speaker was dog or cat and why?
Oh, wow. That's a tough one because I do not have any pets. I would say probably dog. Dogs always seem friendlier. I don't know why that is. I feel like cats are so independent. They don't really need me for anything. But dogs, like to come up and be petted, and I don't know. Maybe I just haven't been around the right cats, but
I just got a cat a few months ago, and she is a Velcro kitty.
Really?
Like, she always is looking for attention.
Okay.
She's really sweet.
Okay. Awesome. We had dogs growing up, so maybe that skews it a little bit. We didn't really have cats. So maybe I just haven't met the right cats.
Okay. Well, thank you so much. It was great to have you. Thank you for all of your words of wisdom.
Thank you for having me, and thank everybody for their questions and for coming out.
All right. Thank you. Yeah.